This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
Hello everybody, good afternoon. Just before we start, I have a quick announcement. So the subject experience survey has been released and will be open until the 3rd of November. Please make sure you complete it. That's very important for us to keep improving the subject, and constructive criticisms are more than welcome. This week we're going to hear about single-cell RNA-seq analysis, and we have a specialist in the house. And I'd like to introduce Dr. Peter Heike from Walter Elisa Hall Institute for Medical Research. Peter is a specialist in single-cell and genomics analysis and leads the bioinformatics analysis of the single-cell open research endeavor and has contributed to multiple papers, methods, and software on genomic analysis, many of which are available at Bioconductor. So Peter was really kind to accept the invitation to talk about single-cell to us. Thank you very much.
No worries. G'day everyone. So I work across the road at the Walter Elisa Hall Institute, or WEHI. It's the Department of Medical Biology here at the university. We have a large bioinformatics research division, I think numbering in around 70 people, as well as bioinformaticians scattered throughout other areas of research like cancer research and malaria. So lots of bioinformatics, and it's becoming more and more fundamental to most biomedical research. About a year ago, we started up a core facility to do single-cell RNA sequencing, single-cell biology, and I started working there as leading the bioinformatics efforts there. So this is what I do day to day. It's a really interesting area and quite a hot area in computational biology and in molecular biology more generally. The technology has only really become available quite recently and has really been an explosion in terms of both technology and methods for analysing these experiments. So at WEHI, we have this group called SCORE, as Douglas mentioned. So we're a team of about 5 or 6 of us where most of the team are wet lab biologists. So they do the experiments, they help develop new protocols for doing these. And then once the experiments are done, I'm analysing the experiments. But it's not just involved, a bioinformatician's role in single-cell RNA sequencing is not just in analysing the data, it's also in designing the experiments. So I'm a statistician by training. I like to ensure that the experiments are well designed so that we can actually answer the questions that we want to do. And I think it's by having a team of not just the wet lab, but also the computational, the bioinformaticians together that you can actually start to do some useful work in this area. And this is my favourite example of what bioinformatics is. My background is statistics. I've come to love biology, and now I see myself in the middle there as a bioinformatician. So you end up being a bit of a hybrid, and you've got to be willing to learn both areas to be able to do your job well, I think. So I think that's the strength we have over at WEHI and our SCORE team, and I'll try and emphasise the necessity of understanding the biology and the techniques when analysing these data as a bioinformatician. So before we get to single cell, I'm going to do a little bit of history on just measuring gene expression. So gene expression is generally thought of as being what RNA is being transcribed in a cell. Roughly speaking, the sort of first high throughput way of doing this, so by high throughput I mean we can measure multiple genes across multiple samples, was in the sort of mid to late 1990s with the development of microarrays. Microarrays allowed you to target particular genes of interest from a pool of cells. So you would take a tissue sample and you would measure the average expression across that piece of tissue, for example. Microarrays developed over the late 90s through the mid-2000s. Basically the development in that sort of 10-year period was that we could measure more genes, we could measure them more cheaply, and we could do more samples. So 10 years of progress, but we're still talking about analysing bulk samples, so a collection of cells and measuring the average expression in those. And these microarrays work by having a oligonucleotide, so a probe on these, basically like a microscope slide. You have your RNA sample and it binds to certain probes based on complementary sequences. So you have to design your probe for the genes you want to measure. So you're limited to studying things that you kind of already know about. But then you can measure how much they are expressed in different conditions. So for example, before and after a drug treatment, or between different tissues, so between a liver cell and a kidney cell. So these worked by having a probe, as I mentioned, and if you've seen these sort of schematic diagrams of a gene before, each one of these sort of yellow boxes is an exon in a gene. Intermediate bit is the intron, so the bit that's not transcribed generally. And the probe would generally, in the early days, only target the 3' end, so the far end of the gene. So all you're measuring there is the presence or absence and how much of the gene, purely based on how much of this part of the sequence is there. Some of the technology over those 10 years was to design probes that captured not just the end of the gene, but we would tile across the entire gene. That allowed you to then say, well, how much was this part of the gene expressed, or was it excluded in certain samples? And so this then brought in basically, as these things got cheaper and cheaper, people started doing larger and larger studies. So early studies would have 1 or 2 samples. Larger studies towards the end of microarrays days were in the tens to hundreds of samples. By the mid to late 2000s, microarrays were largely getting replaced, though, by a new technology called RNA sequencing. So RNA sequencing is taking RNA molecules, converting them to DNA, the complementary DNA sequence, and then sequencing them on one of many different platforms, but high throughput sequencing, which is typified by Illumina sequencing. So this came about sort of the mid-2000s. It really brought about a revolution in terms of the types of questions we could ask, whereas previously with microarrays we could only measure genes that we knew about in advance. With RNA sequencing we could discover what genes we might not know about were ever expressed in a sample. It also allowed us to start looking into species that we might not otherwise have studied. So in order to use a microarray, you needed to have a very good reference, basically. So human and mouse were typically studied. With RNA sequencing, we could start looking at what are called non-model organisms, so kind of the more interesting things often, and really start to... This really accelerated a lot of research in gene expression analysis. But we're still studying populations of cells. So if we took a sample, for example, of blood from one person in this room, and we measured the gene expression, what we would be measuring is the average expression of, say, T cells, B cells, all the different types of cells that make up a standard blood sample. And whilst that's quite useful, what is of greater interest is, well, what is the expression in a particular cell? So, for example, if we took someone who's had a tumour biopsy, we get a piece of tumour. Typically, when that's dissected out, it also contains some healthy tissue. If we used any of these technologies that are on the screen right now, we would be basically measuring what is the expression of the gene in the tumour and the healthy tissue combination. We couldn't tell whether the gene was just in the tumour or just in the healthy tissue. And obviously it's important to know what's actually happening in just the tumour if we want to develop drugs that can affect that process. This is where single cell comes in, and as the name suggests, it's where we can start measuring gene expression in individual cells, and we do that in combination with RNA sequencing. So it's basically a matter of scaling down a technique, RNA sequencing, which was developed for profiling a population of cells that may have had 10,000, 100,000 cells, scaling the technology down so that we could do it in individual cells. And the main limitation is that within an individual cell, there just isn't that much RNA available. So we have to be very careful how we handle it and how we amplify it so that when we measure it, we can be comfortable and confident that what we're measuring reflects what's in the cell and not some artifact of amplifying that sample. So the first single cell study was in 2009. And over the last 10 years, there's been a wealth of new technologies that I'll go through in a little more detail in the next slide. But just to say that in the 10 years of microarrays, from 1995 to 2005, we were basically talking about reducing costs and being able to do more samples. But with the last 10 years of single cell, we've gone from being able to study one cell to being able to study millions of cells per experiment. So experiments that we typically do over at the WeHi will feature a few thousand cells, tens of thousands of cells, but there are projects which I'll talk about that are tasked with identifying, basically cataloguing all the different cells in the human body, and they will profile millions, if not billions, of cells. So this is a figure illustrating that explosion in availability of this technology. So this was the first study back in around 2009, and they did single cell, and they literally did single cell, one cell, and that was a triumph. No one had done this before, but studying one cell tells you what's happening in one cell. It doesn't tell you very much about what it's doing in context of a living, breathing human. And sort of over the next 10 years, each point here is sort of a landmark study in single-cell biology. And you can see that sort of the next study, which was by the same authors, they basically managed to get it up to around 50 cells. Each one of these subsequent ones are different technologies that were developed for doing single-cell sequencing. They're all based on roughly the same idea, is that we need to be able to capture single cells, to isolate them in some way, and then to extract their RNA and be able to label their RNAs so that we know that this RNA came from that cell. So that when we sequence it, we can say, yes, this particular fragment of sequence comes from this gene from this cell. And I'll explain how we do that labeling in a second. This technique here called CellSeq, and its sort of next version, CellSeq 2, is one of the main technologies we use at the WeHi. That's based on physically isolating cells into a 384 well plate. So we use flow cytometry to take a collection of cells and individually drop one cell into each well on a 384 well plate. And then those cells undergo the process of creating RNA sequencing libraries. But probably the most important technology to come out is this one here. Initially this technique called DropSeq, and then this technique called 10x Genomics. So 10x Genomics are a commercial company. And the reason why this is sort of led to a bit of a revolution is that you can go and buy a machine from 10x Genomics that does single-cell sequencing. All these other techniques that are listed here require sort of specialised lab techniques. They require someone in your team, in your lab, who is willing to invest the time to learn it and to develop a very fiddly process. As you can imagine, working with individual cells, there's not much room for error. You're like working with like nanoliters of liquid. Having a commercial kit, which is a box that's about this big, sits on a lab bench, you pop your sample in, it isolates the single cells, it takes the RNA from them and gives you back out a library that you're ready to sequence. And that's really revolutionised the ability of most of labs to use this technique. So just to summarise sort of that history part, initially with microarrays, we're talking around $1,000 a sample, fairly low resolution and fairly noisy. These were using hybridisation, which is a technique that's been around for a long time in molecular biology, but it's a fairly noisy thing. And we could really only study a small number of samples, so N being the number of samples, P being the number of features, so genes. So we were studying maybe 3, 6 samples on maybe 10,000 genes. RNA sequencing, as well as improving the resolution greatly, reduced the cost, reduced the noise, but we're still studying like tens of samples, maybe across 20,000 genes. These days, RNA sequencing is on the order of $50 to $100 a sample, perhaps even cheaper. But this is sort of like prices people were paying commercially a few years ago. With single-cell RNA sequencing, the cost per sample is actually very low. That's because the sample is one cell. But you're doing 10,000 cells, you've got to factor that in, and suddenly it can become quite expensive. It actually has a sort of a lower resolution than our RNA sequencing. it's harder to, we're just working with less materials, so the ability to recover detailed information is a little bit compromised. And it's quite noisy, particularly relative to RNA sequencing. But it finally gets us into a place where we can actually study more samples than genes, for example. We could take 1 million cells and get 20,000 genes from each of those, which, if you've done much in the way in statistics, that's like a nice place to be when you have more samples than you have features that you're interested in. So with this sort of great pace in technology, in 2017, a group came together to make what they call the Human Cell Atlas. So you've probably heard of the Human Genome Project, $3 billion, something like that, to sequence the first human genome. The idea of this is, well, now we've got the genome, we have the sort of the sequence that underlies each cell. Let's create a catalogue of all the different cell types in the human body and what genes they express. So for hundreds of years, biologists have been cataloguing cells in the human body, way back to the days of the invention of the microscope, seeing how cells look physically different from one another down a microscope. This was the idea of, let's make single-cell RNA sequencing our microscope and catalogue what makes one particular immune cell different from another type of immune cell. This is like a multinational effort that's, a lot of the funding is actually through the Chan Zuckerberg Foundation. So Zuckerberg of Facebook infamy has donated quite a bit of money through his organisation towards this project. And over at the WeHi, we're part of this effort to do that. They have a few aims. One is to sort of foster collaboration. But one of the things that they want to develop is what they call the analysis garden, which is a cute little metaphor for developing tools, developing methods to analyse these, to make it so that it makes it easier for everyone in the world to do this. So historically, a lot of times, methods would be developed sort of in-house. They take a while to propagate into the wider research community. They're making a bit of an effort to sort of get these methods out front a bit earlier than they might otherwise be. And so this metaphor, which these slides come from my boss actually, Matt Ritchie, is that ideally we would like the garden to be nice and neat, lovely to walk through. It's very clear what everything is, but in reality, oftentimes the garden of tools to use is an absolute mess. The path isn't clear, and it can be quite daunting to know where to begin. So what we're going to do, hopefully in this lecture and in the lecture on Friday given by a colleague of mine, is to sort of explain some of these tools and give you ideas on methods that are out there and the sort of challenges that are there as a bioinformatician and resources where you would might learn more about analysing these types of data. So I've shown you that the technology has exploded. We've gone over the last 10 years from sequencing 1 cell to 1 million cells, if not more. And accompanying that has been an explosion in the number of software tools and statistical methodologies for analysing these data. So this is only going back to 2017. So within 2 years, there was around 50 tools about 2 years ago. This data is being updated constantly. Some colleagues down the road, Luke Zapier, has developed this database of tools. And as of this morning, there are some 500 tools available for analysing single-cell data. As someone like myself who specialises in single-cell analysis, I can tell you I know about 10, and that this is just a completely unsustainable kind of growth. And so it becomes important to develop benchmark strategies to figure out what tools should we be using. And so there's been a lot of effort to make benchmarking experiments where you know the truth beforehand and you apply these different tools to see what methods recover the truth and what methods sort of give you nonsense. But it's still quite a daunting process. There's lots of different types of analysis you can do with single-cell data. Almost 50% of the tools are just for visualising the data. And that's because it actually becomes quite challenging to think about. Basically, what we do when we do single-cell RNA sequencing is we measure gene expression on individual cells. So if you think of your data set as a matrix, each column of your matrix is one of your cells, and each row is a gene. We might have a matrix with something like 10,000 columns, so 10,000 samples and 20,000 genes. And trying to visualize that 10,000 by 20,000 dimensional data set in a 2D picture on your laptop is quite challenging. And even more challenging is to do it efficiently and in a way that best represents the structure of the data. So there's been a lot of effort towards that. Clustering is the idea that we want to take the RNA sequencing data and find what cells look like other cells. So can we cluster these cells together and then say something more broadly about those clusters as a whole? And there's a whole raft of methods developed for that. The point here is to say that for each of these very important steps, there are multiple things that can be, multiple options available. And to sort of get started on that, you need some guidance. Finally, just on the tools front, if you are a sort of more on the computational lens, most of these tools are written in the R programming language, which is very popular within bioinformatics generally. It's very popular within statistics as well. Python is also popular. They're the two main ones, sort of data analysis type languages. R is in large part very popular because of the bioconductor project, which has been around for about 15 years now, I think, 10 to 15 years anyway, of which is a collection of packages for analysing biological data. So things like gene expression from microarrays, from RNA sequencing, chip sequencing, attack sequencing, any of those types of things, there's generally an R package in the bioconductor project that's available for it. So most of what I'm going to talk about, the tools I talk about today, are actually in R. That's the main language I use. I do use Python. I use C++ when I need to, but first and foremost, I use R for this sort of work. So where to start? This is my basically main recommendation. If you want to analyse single-cell data, you need to analyse single-cell data, particularly if you're already interested in R. There is a book. An online free book called Orchestrating Single-Cell Analysis with Bioconductor. It's an ebook, free. It's been constantly updated with new sections based on like new technologies, new methods that are developed, and it walks through all the steps of what are needed for a single-cell analysis. It also includes a pretty good introduction to R and to Bioconductor, so if that's something you still feel you need a little bit of help with, it might be a good place to have a look. I've been doing bioinformatics for 10 years or so now, and this is still like my daily reference for what I'm doing in my day-to-day work. Another great place to start is this review article that's open access, so you can get it without having to go through all the library shenanigans, which sort of explains the current best practices as a bioinformatician for analysing single-cell data. They take a sort of, I guess, a software agnostic viewpoint. They talk a bit about R, they talk a bit about Python, and walk through the sort of conceptual ideas of what needs to be done at each stage and point out what has been done in the literature to show that this is the best method for that particular step. So this is sort of an illustration of the analysis process for single-cell RNA sequencing. So it starts with sequencing the individual cells and getting the FASTQ files. The FASTQ files need to undergo QC checking and the like, alignment to a reference genome, counting the number of reads at each gene to get what are these count matrices. Quality control is critical. And I'm going to spend most of my part of this talk talking about quality control because these data are very noisy. There's a lot of scope for technical biases, things that may have gone wrong during the experiment. And you're basically wasting your time doing the rest of the analysis if you haven't done your quality control very well. And generally in conjunction with the biologists, if it's not you, to help so that you understand what they want to get out of this experiment and any particular oddities of the biology they study. Normalization, I'll also talk about today, and then Davis on Friday will talk more about the downstream analysis, which is trying to actually learn about the biology of the cells from these experiments. So that includes doing these clustering that I talked about, as well as doing things like differential expression and looking at gene networks. So we're going to go through this analysis garden, starting with the FastQ files. Along the way, I'm going to highlight a few bioconductor packages. So these are illustrated by these stickers here. Bioconductor like to make stickers for their packages, so you can put them on your laptop and show off what you've been working on, basically. So along the way, I'll highlight a few of these, but I've also tried to include some things for non-R audiences along the way. So, single-cell sequencing. We need to get single cells, first of all. So, for example, say we're studying liver. Liver is the cells do not sit separately from one another. They're all interconnected, and we need to dissociate or separate these out into individual cells. Now, this is a pretty easy process for something like studying blood samples. You take a blood draw, blood is a liquid, things are floating around, it's fairly easy to isolate individual cells. But when you're working with a tissue, like brain, liver, this is the first step, and this is like a lab technique. There are techniques for separating tissues out into their individual cells. It's important to bear in mind that that process can actually change gene expression, so you need to be aware that simply by taking your tissue out of its natural environment and then separating it out, you will change the gene expression of cells. Unfortunately, there's generally no way around that. It's just something you've got to live with. Single-cell isolation is actually two sort of main ways. The first is on the CellSeq technology that I mentioned, where we have these 384 well plates, and we use flow cytometry, which is a molecular biology technique that enables us to get individual cells and basically pop one cell in each well. So they are physically isolated from one another on a standard plate that's in any molecular biology lab. The 10x genomics platform uses a neat technique where they have oil-water droplets. So basically, if you ever put oil on top of a water surface, you know that they'll form little beads where the oil beads will not contain any water within them, for example. By having sort of two streams of oil and water, or I'm not sure if it's actually water, but a liquid of some sort, and then shooting cells in at a third direction, you can capture an individual cell within an oil droplet. Within that droplet, you also capture the molecular biology reagents that are necessary to reverse transcribe the to first lyse the cell, to burst the cell, so that the RNA is available. It's all captured within this droplet. Then within that droplet, there's also the molecular biology reagents to turn that RNA into cDNA, the complementary DNA sequence, and add these following steps, which I'll explain in a moment. But it all happens within these droplets, which is quite neat and is what 10x Genomics have been able to successfully commercialise. So once the cells are isolated, be it by sort of physical isolation in the plate or by droplets, the RNA needs to be accessed by lysing the cells and then converted to DNA, or complementary DNA, using reverse transcription. We then need to amplify that cDNA. Within an individual cell, there's not much RNA, there's not much cDNA to work with. These sequencing technologies require on the order of nanograms or picograms of DNA, so we have to amplify that. And traditionally, that's done by PCR, polymerase chain reaction, which is a workhorse of molecular biology. Another technique that's sometimes used is in vitro transcription, IVT, followed by reverse transcription. So two different ways, but the point is to amplify up the pool of cDNA and then to add on the necessary sequencing adapters so that it can go on the sequencing machine and be sequenced. So that is to get it onto the sequencer. And once it's sequenced, we end up with our FastQ file, which you've probably seen, which has a read ID, the sequence, and then some quality information. And then we need to get a table of counts saying how much of each gene is expressed in each sample. So these files will have hundreds of millions of rows. With most sequencing experiments, you want to do some sort of initial QC of those. A popular tool for that is FastQC. It will enable you to look for contaminants, so things that aren't related to your sample. So they might be, what do you call them? Barcodes, amplicons that have been contaminating your sample. You can look it against the database and find those and then discard those reads from further analysis. What is sort of key to doing these is single cell technology is that we are able to label every RNA molecule with 2 barcodes generally, 1 or 2 barcodes, but generally 2. So the first barcode tells us what cell this sample came from. And the easiest way to explain that is for the cell seq. So when we have physically isolated the cells onto wells of a plate, within each well we have adapter sequences which contain a known sequence, typically on the order of 8 nucleotides in length. So we know that in the top left position of this plate, we know that the sequence is ACGT, CGAC, something like that. When we do our reverse transcription, these adapters are added to our mRNA molecules. So now we have the gene as well as a known sequence that tells us what cell that sample came from. We also have these things called molecular barcodes, or UMIs, unique molecular identifiers. These are actually random sequences. We don't know ahead of time what these sequences are, and they are also within each well on the plate or within each droplet in the 10x platform. What they enable us to do is that they are added to the mRNA molecule so that when we amplify it, if we sequence the amplified sample and we find 2 things that have the same gene sequence and the same molecular identifier, we know that's just a PCR duplicate. We've just copied that same molecule again, and so it's kind of redundant information. Whereas if we find the same gene sequence, but it has a different molecular identifier, we can infer that in fact there were 2 copies of that molecule, 2 distinct copies of that molecule to begin with. So it enables us to separate out during this amplification process whether 2 copies have arisen because there were 2 copies to begin with, or because we've just repeatedly copied the same molecule over and over again. So these sequencing reads conceptually have a sequence that relates to the gene of interest, a sequence that is known to us in advance generally, that tells us what cell or what well on the plate these samples came from, and these molecular identifiers that enable us to say whether it is a copy of the exact same mRNA or if it is 2 distinct mRNAs. So this UMI idea, these molecular identifiers, aren't typically used in ordinary RNA sequencing. So there has been a raft of methods developed to deal with these. So what you need to do is for every sequencing read, you need to identify what bit it's typically known like the first 6 base pairs might be your cellular barcode, the next 8 base pairs are your unique molecular identifier, and the remaining sequence is the gene of interest. But sometimes that's a little bit offset, so it might be bases 2 through 7, 8 through 15. In another read, it might be 3 through 8, etc. And so these tools were developed to quickly go through these FASTQ files, which contain hundreds of millions of reads, and quickly separate it out into which bit is the cell ID, which bit is the UMI, and which bit is the transcript ID. They also need to account for the fact that sequencing isn't perfect. When you sequence DNA molecules, there are errors, and we want to be able to correct for these errors. And so they often have methods for doing error correction. These might be as simple as saying, well, here's our database of known barcodes. Here are the barcodes that we sequenced. Find us the nearest match. So we might say we will tolerate anything up to 1 mismatch between a barcode and what we think should be in the database. To do that quickly is actually kind of an interesting computer science problem, and there's been quite a number of methods developed for it. I just mentioned this software SC Pipe, which we use for analysing a lot of data over at the WeHi. It includes that as part of it. So SC Pipe is sort of an end-to-end analysis pipeline, and one of those steps is doing the UMI handling. So we have to extract the cell barcodes and the UMIs, do some correction, and then we need to take the RNA molecule part and identify what gene it came from. And so in this case, it's basically like doing RNA sequencing. We need to align it to a reference genome or a reference transcriptome. So to do that, we could use any one of a number of tools. Popular ones are STAR, which is an RNA-seq aligner that can account for the fact that RNA molecules are spliced. So a typical DNA sequencing aligner isn't the best choice of tool for this problem. More specialised tools such as STAR is generally slightly better. There you're mapping RNA against your reference genome. So that's how these ones work, and then you end up with your BAM file, which is your file of aligned reads, and then you need to count basically how many times each gene was detected in each sample. So you need to go back to your barcodes and figure out this read, which sample did it come from, and then add a count to the count matrix accordingly. There's also these other class of methods for doing this step, which are called pseudo-aligners, the most popular of which are Callisto and algo20. So the main point of difference between Callisto and algo20 and these first three, like BWA or Bowtie and STAR, is that Callisto and algo20 only map to the transcriptome. So they only map to the genes, basically, rather than the full reference genome. So for example, human reference genome, 3 billion bases. Most of it are not genes, they're intergenic regions. These other methods just say, well, these are RNA molecules. They should have come from the genes part of the genome. Let's just map against those. They also have a few other tricks that mean they don't do a traditional sequence alignment like is done in Bowtie or BWA or like in BLAST, where it might actually do like a full Smith-Waterman alignment. They use things like k-mer matching, which is a fast way of identifying sequences against a reference, against a database. And the main advantage of these is that because they're only mapping to the transcriptome rather than the reference genome, plus some other computational tricks, is that they're a lot faster. And that can be quite advantageous when you're processing millions of cells. So these have become more and more popular as the ability of people to generate lots of samples has increased. So SC Pipe, as I mentioned, is one software that I use a lot of. The steps that we sort of talked about there were processing of the FASTQ files, the alignment of these reads to the reference genome, counting which exon each read came from, or which gene each read came from, and then figuring out what cell barcode matches that read, and then adding all those things together to get a count matrix where each column is a sample and each row is a gene. This is one way of doing it. There are others. SC Pipe is particularly good for the CellSeq platform, and that's where I use it most of all. The nice thing it also has is that it outputs sort of the standard data structure that is used within the R and bioconductor worlds for doing single-cell analysis. And it's a data structure called a single-cell experiment. So that just means I've got nice interoperability with other tools in the sort of garden of tools. It also does some QC analysis, which I'll explain in a moment. The single-cell experiment is the sort of core data structure. And it's broken down into a few different parts. So the first one is this in blue, is the count matrix. So each row here is a gene, each column is a sample, and here's an illustration of some data that might be in that there. So this would be saying that for the first sample, the first gene was not observed, 0 counts. For the second gene, we observed one count, whereas in the second sample, we observed 2 counts. So that's the data we want to analyse. It's the counts, how much each gene is expressed. But there's additional information available for each sample. For each row of this data set, each row is a gene. We might have the ensemble gene ID. We might have other information about that gene, like which chromosome it's on, whether it's a sex chromosome, whether it's on the autosomes, because sometimes we want to be able to filter out things that are, for example, on the mitochondria. Often not interesting for downstream analysis, so we want to be able to identify rows that are from particular chromosomes. And then we also have information on each cell. So is that cell from our treatment or our control? Is it from person 1 or person 2? Is it from the gene, is it from the knockout or from the wild type sample? And so this data structure is sort of has been agreed upon by people in the R bioconductor world as this is the way we're going to represent all that information in a consistent manner, so that if I'm developing a tool to analyse single cell data, I know that I can expect that the rows are genes and the columns are samples, and then if I want to find out about information about the samples, I can use this column data function. If I need information about the rows, I could use the row data function. And it just helps people who are developing these tools interoperate with each other. So the count matrix itself, one of the main characteristics is that most of it is 0. 60, 70, up to 90% of the counts are 0. And that's because there is, as I said, there's very little material within each cell to start with. So oftentimes we lose that material along the way. These are often called dropouts, where a gene may have been there, may be expressed in the cell, but we failed to sequence it, we failed to measure it accurately. It's also because when you're sequencing 1 million cells, you have to do a lot of sequencing, and that becomes quite expensive. So often people will only sequence relatively shallowly. So they don't get, per cell, they don't capture the full information. The hope is that by sequencing lots of cells shallowly, they can learn more than if they just studied a small number of cells in great amount of detail. So the count matrix of genes by samples will mostly be 0. For any given cell, any given column, you typically find on the order of 1,000, 2,000, 3,000 genes have a non-zero count. And we consider that the gene being expressed. So given that, for example, the human genome is roughly 20,000 genes, per cell we're only capturing maybe 10, 5% of genes available. Also, by column, different cells will be sequenced to different depths, perhaps 100-fold differences in sequencing depth. And that just really reflects the random variation in the sequencing process. All this is to say is that the count matrix is very noisy. There's a lot of technical factors that affect this, and that we need to do very careful quality control on these. For example, if a cell has almost every single count as 0, then we may as well throw it out. There's nothing to be learned from that cell. It's going to complicate any statistical methods downstream. And so that's what our QC needs to account for. We also need to do the opposite of, like, is a gene expressed in any sample? If the gene's not expressed in any of our samples, then we can throw it out and simplify our lives. So the quality control is basically about removing cells by removing columns of that matrix, which are samples, or rows of that matrix, which are genes. So some standard filters we might apply is the library size. So that's how many molecules were sequenced from that cell. We might discard any cell that has, say, fewer than 100 reads. There's very little we can say about a cell when we only measure 100 reads across the genome. But this needs to be influenced by the data itself. So for some samples, if we sequenced it very deeply, then we might say, well, we require at least 1,000 reads. Conversely, if we knew we sequenced very shallowly, we might say, well, we'll accept anything with at least 10 reads. So the choice of cutoff has to be influenced by how the experiment was done. Similarly, we might filter out based on how many genes were expressed in each cell, as well as other technical factors which are specific to the type of assay you're doing. But, for example, mitochondrial gene expression is often associated with cells that are stressed, that might be dying. So the process of doing a single cell experiment can kind of traumatise the cells. So if you find a sample where lots of the expression comes from the mitochondria, that's often a good reason to throw that cell out, because it may indicate that this cell has been so stressed by the process that what you've measured does not really reflect what you're interested in studying. We then might also, after having removed the bad cells, we might remove genes that are of no interest. I don't do much in the way of this. I typically only remove genes that aren't detected in any cell. Other people might require the gene to be detected at least in a certain percentage of cells. So SC pipe gives you some quality metrics, for example. Any of these software pipelines will give you things like this. So I'll explain this one a little bit as an example. Each column here is a cell, so there's several hundred, if not thousands of cells. And what's shown is what percentage of reads were unaligned, aligned to the aligned but not mapped to a gene, where we have an ambiguous mapping, so it mapped equally well to different parts of the genome, whether it mapped to these control regions, whether it mapped to introns and mapped to exons. First of all, you can see for this sample, only about 25% of the data actually was successfully mapped to exons. Now, 25% is not like a standard number, but just to give you an idea, like we've spent a lot of time sequencing. 75% of things we've sequenced aren't going to inform us about gene expression. And this is pretty typical. It's a very noisy experiment, and while you'd like to see more than 25%, this is not atypical. You can also see a subset of samples down here where they basically have no data coming from the exons. So these are the type of cells that we'd want to remove in our QC analysis. So any sort of process, any sort of software you use will give you these sort of metrics, and you need to look at these and decide where the cutoff should fall. Generally, you can just filter based on these cutoffs, but you also do need to consider the biology of these cells. Just as an example, if these two plots were overlaid with one another, this is the total number of reads sequenced versus the number of genes detected, and each dot is a cell. We might discard these cells down here because they have small total counts. In fact, these come from a particular type of cell, in this case, for this experiment, quiescent cells. So quiescent cells are ones where they're not doing much, they're not transcribing much of their RNA. So you need to be careful that your choice of cutoffs doesn't remove cells due to the biology of that cell. And that requires talking with or understanding the biology of the system well enough that you can make those decisions. 10x's software Cell Ranger is used for processing their droplet-based single-cell data. And one of the QC plots they give you is this one here. Each point is one of their cells, and it's it's the library size. How many molecules did we detect? So the most sequenced cell we got on the order of 50,000 reads from. And what we look for is, as we plot this, is where does this drop off? Where do we start not getting many reads coming from? And the trick there is to find this point here, because basically the remainder are ones where the droplet actually didn't contain a cell to begin with. We've sequenced just some junk that was floating around. And the reason I highlight this one is that 10x is a commercial company. They had a method for doing this for solving this problem of identifying empty droplets. But then some colleagues of mine came up with their own method that they showed was much better than 10x's method. And 10x was like, okay, that's a good point. And then they integrated that into their software. So they have taken lessons from research and adapted them to make their products better. And I think that's a really nice example of where bioinformatics can help in that regard. Finally, for QC, this package here, Skater, which is on Bioconductor, is sort of the go-to for QCing most single-cell data sets, I find. It has lots of useful plots and ways of identifying outlying cells. Just finally, normalization is critical. So normalization is so that if we have two cells that are basically the same, but we sequenced one 100 times more than the other, it will look like initially that it has 100-fold higher gene expression of every single gene. Normalization is to basically divide through the counts by the library size. For bulk RNA sequencing, that's a pretty well studied and pretty well solved problem. For single cell data, it's a little less clear what's the best way, although a recent paper has shown that this method of SCRAN, which is another bioconductor package, generally performs quite well, and that's, in my experience as well, is good at normalizing. So that's sort of the first part of the single cell analysis, and a lot of it is on doing QC. The next parts are where you actually start doing the interesting biology, but it's all for nothing if you've not done a good job on your QC process, and Davis will talk about those next steps, things like dimensionality reduction and downstream analyses, and talk about some of these packages that I haven't talked about today for doing so. So single-cell RNA-seq, it's a very powerful technique developed over about the last 10 years for measuring gene expression in individual cells. It's a very noisy experiment. There's a lot of technical noise, so careful QC and statistical analysis is required. And the QC is to remove bad cells as well as genes that are sort of uninteresting. If you're interested in learning more about these, the first reference is one I gave at the start, which I highly recommend. A few others, including courses and a sort of 1 hour version of the entire process that I gave recently at a workshop. What I've shown you today is actually, most of this is from my boss, Matt Ritchie, and with contributions from Yunshun and Saskia and Luyi, who also worked at WeHi. So thanks for your attention, and if you have any questions, I'll be around for a bit if you want to ask.
I have a question.
Yeah.
So over the 10 past years, a lot of attention has been drawn to single cell. Therefore, we have what you said, 500 different methods. Do we still need more methods, or just now it's just a matter of navigating through the garden and finding the right one?
Yeah, I'm sure there'll be need for new methods because the molecular biology is also advancing. So if single cell is now, not only can we measure gene expression from individual cells, we can separately extract the DNA and the RNA and measure both of them from an individual cell. So correlating, for example, mutations in the DNA with expression changes from individual cells, that will need a whole new raft of methods. Or measuring transcription factor binding via a ChIP-seq type thing on the DNA, or chromatin accessibility using attack sequencing and relating that back to gene expression. Then there's also just, if you're more on the sort of computer science end of research, of making these methods more computationally efficient, because oftentimes these methods are developed and they work. You can show they work and they're great, but they work really well when you've got 1,000 cells. Now you've got 1,000,000 cells and they don't work so great. So scaling those up is still an important part. But probably more important is than making new methods is making documentation and workflows for how to use these methods, because it's no good making a method if people can't use it.